Hello everyone, in this video we are going to look at a case study of purification and characterization of a novel bacteriocin produced by Lactobacillus plantarum strain J23 isolated from the Chinese fermented milk. So, in China, a lot of fermented vegetables, meat and milk is being consumed since ancient times. These fermented food contain some bacteria which produce bacteriocins and inhibit the growth of food spoiling bacteria and thus increasing the shelf life of these foods. Bacteriocins are proteinaceous compounds which is produced by one bacteria to limit the growth of other closely related bacteria to reduce the competition for survival. Also, bacteriocins from lactic acid bacteria have a potential application as natural food preservatives in the food industry. So, starting with the inoculation of Chinese fermented milk on MRS medium, whose composition is as follows. After incubation, 300 bacterial isolates were obtained and then each isolate was again inoculated in MRS broth separately. After incubation, tubes were centrifuged to obtain the pellet and supernatant. The supernatant of each isolate is now used to perform the agar well diffusion assay. This assay is done to check which isolate produced bacteriocin. In this assay, an agar plate containing an indicator bacteria, for example here they have taken Listeria monocytogenes, is used. Wells are bored in this plate and the supernatant obtained from each isolate is inoculated separately in each well and kept for incubation. After incubation, if clear zone is observed around the wells, that means the supernatant of that particular isolate contained bacteriocin and thus it inhibited the growth of Listeria monocytogenes, while if no clear zone is observed around the wells, that means no bacteriocin is produced. The results that they got are as follows. Out of 300 bacterial isolates, only 6 isolates showed antibacterial activity in the agar well diffusion assay, out of which J23 strain showed the best result than the rest 5 strains. Therefore, J23 was selected as a potent bacteriocin producer for further research. The biochemical characterization of J23 strain revealed that it was a Lactobacillus plantarum. Its optimization was done to get the maximum bacteriocin yield. Further, the extraction, purification and identification of the bacteriocin was done by using different methods which are as follows. So, starting with the first method, ammonium sulfate precipitation. The optimized culture of Lactobacillus plantarum was centrifuged, supernatant was obtained and then different concentrations of ammonium sulfate was added in the supernatant, that is 40, 50, 60, 70 and 80%. Due to this, the proteins precipitates, and since bacteriocin is also a protein, it also precipitates. The mechanism behind it is that ammonium sulfate competes with the protein molecules to bind with the water molecules. This reduces the ionic strength between the proteins and the water molecules. Thus, protein solubility in the water decreases, resulting in its precipitation. These precipitates obtained by adding different concentrations of ammonium sulfate were collected, dissolved in distilled water and used to perform the agar well diffusion assay. The results are as follows. As you can clearly see in the image, precipitate obtained by 80% ammonium sulfate showed the largest clearance zone, indicating that 80% ammonium sulfate when used gives the maximum bacteriocin yield and thus the maximum antibacterial activity. So till now the precipitate that we have got is impure, that is it contains bacteriocin plus other proteins also. So to get pure bacteriocin, the next two purification techniques are used, that is cation exchange chromatography and reverse phase HPLC. Cation exchange chromatography. In cation exchange chromatography, the principle is that there is interaction between the oppositely charged components in the stationary phase and the mobile phase. Here a column is filled with resin with negatively charged groups on it, which is the stationary phase, and the mobile phase is a phosphate buffer solution of pH 6. First, the column is equilibrated with this buffer so that cations in the buffer bind to the negatively charged groups on the stationary phase. Then the sample is introduced in the column. The sample is that precipitate obtained by 80% ammonium sulfate in the previous method, dissolved in the buffer solution. The negatively charged proteins in the sample flows out of the column due to repulsion with the stationary phase, while the positively charged proteins binds to the negatively charged groups on the stationary phase by replacing the cations in the buffer which were earlier attached during equilibration step. Therefore, this method is also called as cation exchange chromatography, since cations in the buffer are exchanged by positively charged proteins. Then the elution of the positively charged proteins is done by using 1 molar NaCl. NaCl reduces the ionic strength between positively charged proteins and the stationary phase. Greater the positive charge on the protein, later it will be eluted. All eluents are collected separately and quantified by UV absorbance and perform the agar-well diffusion assay. The results are as follows. As you can clearly see in the graph, eluent collected at 650 to 740 ml showed the maximum antibacterial activity in the agar-well diffusion assay, indicating that it is the bacteriocin. Also, another piece of information that we get is bacteriocin is highly cationic since it is eluted very much later, and its concentration is also very low, indicated by the UV absorbance on the y-axis. The next purification technique used is reverse phase HPLC. Reverse phase HPLC. In this, separation of components is based on their hydrophobicity or polarity. A column containing silica particles bonded with C18, that is 18 carbon long hydrocarbon chain, is the stationary phase, while the mobile phase is a mixture of organic solvent and water. The sample is injected in the column by the mobile phase. Sample is that precipitate of the 80% ammonium sulfate. Initially, the ratio of organic solvent to water is low, hence the hydrophilic molecules elute by the mobile phase early, and the hydrophobic molecules remain attached to the stationary phase. As time increases, the ratio of organic solvent to water is gradually increased, and hence now the hydrophobic molecules which were attached to the stationary phase now enter in the mobile phase and elute later. The following time and gradient was used. For the first 20 minutes, ratio was increased from 0 to 60%, then the next 5 minutes 60 to 100%, and in the next 5 minutes 100% ratio was kept constant. All the eluents were collected separately at different time intervals and used to do the agar-well diffusion assay. Results showed that the eluate collected at 24.9 minutes showed antibacterial activity, indicating that it is the bacteriocin. Also, the bacteriocin is slightly hydrophobic since it is eluted in the later stage. Now, since we have the purified bacteriocin, now it is time for determining its molecular weight, chemical structure, etc. For determining the molecular weight of the bacteriocin, SDS page is used. SDS page is a technique in which the separation of proteins is based on their size. It has the following components: gel matrix on which the proteins separate. It is a combination of stacking gel and the resolving gel. Both have the same composition, but the only difference is in their pH. Stacking gel has a pH of 6.8 while the resolving gel has a pH of 8.8. Next, there are electrodes for generating electric field, that is negatively charged cathode and positively charged anode. The next component is our sample, which is a mixture of proteins to be separated. Beta mercaptoethanol, which breaks the disulfide bond in the protein, making the protein chain linear. On this linear chain of protein, SDS binds to give a uniform negative charge to the protein. One molecule of SDS binds to 2 amino acid residues. Due to this, the charge to mass ratio of each proteins becomes same, and now the proteins will be separated only based on their size and not charge. Bromophenol blue is used as a tracking dye to visualize the movement of proteins. Glycerol gives viscosity to the sample. Next component is the running buffer, which is trisglycine chloride of pH 8.3. Lastly, Kumasi blue is used to stain the protein bands. The procedure is as follows. Firstly, the sample is introduced in the wells. The first well is always a protein ladder which has different proteins of known molecular weight. Negatively charged proteins in the sample and the glycine and chloride ions in the running buffer start moving towards the anode. Their motility rate in the stacking gel is chloride ions, then proteins, then glycine. Therefore, proteins get stacked between the chloride ions and glycine so that all the protein reaches the resolving gel at the same time. After entering in the resolving gel, motility rate changes as follows: chloride ions, then glycine, then protein. Since glycine becomes more negatively charged at higher pH and moves faster than proteins. Among the proteins, small proteins will move fast while large proteins will move slow, creating protein bands at different distances in the resolving gel, which are then stained by Kumasi blue for visualization and comparing with the protein ladder for determining the approx molecular weight of each protein in the sample. Let's see the results. Since our sample contained only one protein, that is the bacteriocin, only one single band was observed between 4.6 to 10 kilodalton. On careful examination, the molecular weight of bacteriocin was found to be 6.73 kilodaltons. Next, LCMS was performed to determine the chemical structure of bacteriocin. The active band from the SDS page gel was analyzed with the LCMS method, and it revealed that it was a novel bacteriocin, and it was named as bacteriocin lac B23. So in this way, following a series of separation and purification techniques, a novel bacteriocin was isolated from Lactobacillus plantarum found in the Chinese fermented milk. If you want to read more details about this article, you can visit the Frontiers website, link is given here. So that's it for this video. Thank you so much for watching.